This is the third part of the video key for the genetics presentation. This part will cover sex-linked traits. Remember when you do sex-linked problems that you're going to have to keep track of your sex chromosomes, the X's and Y's. You also need to remember that Y chromosomes are teeny tiny and they don't carry many traits. Therefore, they are naked. All of these traits are going to be carried on the X chromosome. So in fruit flies, we have eye color sex-linked, and red is dominant to white. So first step is to set up a key of alleles, just like any other problem. So big R is dominant, so red, little r is going to be white. Our male fly is white-eyed. This means that he has X, little r, Y. The female's genotype is homozygous dominant. So she's going to be X, big R, X, big R. So once we have our cross, we can plug this into a Punnett Square. Remember to keep track of your X and Y chromosomes at all times. So dad is X, little r, Y. Mom is X, big R, X, big R. We can plug this in. Even in our Punnett Square, we're going to have all of these X's and Y's. So now we need to go back and answer what is asked. So this question was asking for the phenotypic ratio of the offspring. So remember that the gender is part of your phenotype. So in this case, we have two females, and they are both red-eyed. So 50% red-eyed females. In these other boxes down here, we have two males, and both of them are also red-eyed. So 50% red-eye male. Be sure when you're doing these problems that you give me the full phenotype as well as the percentage or fraction. So hemophilia is a sex-linked disease that we see in humans. First, we set up a key of alleles. Our big H, dominant, is going to be a healthy individual. Our recessive, little h, is going to be the one with hemophilia. In the problem, it says a man who is normal, so no hemophilia. This means that he is X, big H, Y. And he's going to marry a woman who is a carrier. So remember, carrier is just another word for heterozygote. So it's referring to her genotype. So she's female, she's X, big H, X, little h. So remember to keep track of your X and Y chromosomes when you're putting these gametes on the Punnett Square, and to keep track of them when you're filling in the Punnett Square as well. So our first box is X, big H, X, big H. Second box, X, big H, X, little h. We have X, big H, Y, X, little h, Y. To answer the question, we need to report the phenotypic ratio of the children. So translate what is in each one of these boxes. So if we look at the top two boxes, we see that these are both females, and they have at least one copy of the big H allele, which means they are both normal, healthy individuals. So we have 50% females who are healthy. When we look at the bottom boxes, we see that there is one male with a copy of the big H, so he is healthy, so 25% male and healthy. The other box has a male with a little h. So this means that we have 25% that are male with hemophilia. So the second part of this problem is asking about the chance of their son having hemophilia. So because we're just talking about their son, we're going to focus on the bottom two boxes. So we know that they have a son. So out of those two possibilities, one of those boxes has a child with hemophilia, which means that there's a 50% chance that their son will have hemophilia. So this would be a little bit different if I asked you what was the chance of a child having hemophilia. So the chance of any child having hemophilia is 25%. So at that point we're looking at the whole Punnett Square again, not just the bottom two boxes.